In this video we will discuss about a new topic, or we can say different topic for this channel, that is micropattern soft gel to study T cell response to mechanical properties of antigen presenting cells. So in vitro it is difficult to understand the interplay between T cells and the antigen presenting cells because of this mechanical changes that occurs between T cells and antigen presenting cells interactions. So this paper had designed a hydrogel where they can control the biofunctionalization of the substrate in a way that they can study the T cell response to different T cell receptors on the hydrogels. So what they have done is that they have used this soft substrate and stiff substrate, and first they, in one of the case, they analyze this soft substrate and use the T cells to see how the mechanotransduction occurs without any biofunctionalization with any T cell specific receptors, and this is what they have observed. And in other case where it is much more complicated, where what they did is they use the stiff substrate and biofunctionalize it with anti-CD3 and check whether there is any changes occurs in the T cells or not. And what they have find out, there is a changes that occurs in the ZEP70 phosphorylations and F-actin. This is enhanced, and this is enhanced in the stiff substrate, but these pathways are less enhanced in the soft substrate. We will discuss that thing in the subsequent videos. And here they are able to control this ligand density and even define the mechanics, that is they are able to control the ligand density with control with having control the stiffness of the substrate. And this is what the high stiffness they found, the effectins is changes, and the ZEP70 phosphorylations are also increased. And these things they have observed using this thing they have observed when they biofunctionalize this substrate with the anti-CD3, and these anti-CD3 are being interacted with these T cell receptors or CD3 receptors that are presented on the T cells, and those T cell receptors will cause change in the actin filaments in the T cells. And in comparison so to this stiff substrate, when they move to the soft substrate, what they have find out is that there is not much changes that occurs in the ZEP70 phosphorylated ZEP70 that is already phosphorylated. But what they have observed is that the results that they are getting with their micropatterned soft hydrogel that they have developed is similar to the results that people have seen in the in vivo studies of the T cells interactions with the antigen presenting cells. So by comparison, these in vivo results and their micropatterned soft hydrogels, or this micropatterned hydrogel with a control on the stiffness, and they are able to show that their in vitro developed their in vitro developed this substrate is able to replicate the results that are seen in the in vivo. So this is, they have just shown just one example about this anti-CD3 receptors. We will see in the paper whether they have multiple examples or not, but from this they are able to point out that one can use different biofunctionalized substrate to biofunctionalize ligands to, or one can use different ligand to biofunctionalize this hydrogel so that they can study the interactions of their cells of interest against those ligands that are biofunctionalized on the substrate. So this will cover how they have developed their substrate and what are the changes that they did in their experiments in order to get the results that is similar to the in vivo observations. This is just a graphical abstract that we have covered in this video. We will cover their experimental results in the subsequent videos. That's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.